Okay. Am I not audible loudly? Hello?
Now you are audible.
No, it's
Now you
perfect.
are audible, ma'am.
Audible. Okay, okay, thanks. So I have started the recording. Okay. So let's begin today's class. So in previous class we actually talked about a tissue repair process. What are the steps in the wound healing that we have already seen? But I also checked your guys quiz 2 paper. Okay, and there I asked you you have to define what is transdifferentiation, and there I saw many of you have written that trans differentiation is the change in the cell type from one to another, but also mentioned casually this term metaplasia. So I think many of you have copied the answer from Google, because that is the first answer that you get when search transdifferentiation. So if I don't know how you have written the term metaplasia, because I have not discussed it in class, but I thought it's going to be very good actually if I discuss very briefly the difference between these three terms: transdifferentiation, metaplasia, and plasticity, because these three terms are used are having very similar meaning, but they are slightly different from one another. Okay, so transdifferentiation, if you recall, uh, here transformation of a differentiated cell type takes place to, and it becomes another differentiated cell type, like a fibroblast can transdifferentiate if given proper environmental cues to become, say, a chondrocyte. Okay, so this is transdifferentiation. Now direct, why I'm using the term direct, because there is no intermediate cell stage. Okay, so there is no stage, intermediate cell stage, where the cell has become immature or have gained stem cell like characteristic or progenitor cell like characteristic. It is directly changing its morphology and properties, or the cell properties. Whereas metaplasia, here this term is used to define the conversion of one tissue type to another. Now, metaplasia is actually a very big term. It is used in various contexts. Even before, like, development of cancer, this there is a metaplasia state that happens. So metaplasia doesn't always guarantee the development of cancer, but uh, initial during the initial stages metaplasia is observed. So here it is used to refer to any switch of cell type regardless of pathway. Okay, so here cell type is changing, and it is this term broadly, very broadly, used to define any type of cell type change. It also includes interconversions between stem cells. So one type of stem cell can change and become another type of stem cell. It can also include the case where a small population of cell type, which is in minority, it is minor cell types, they can selectively outgrow, means they can start proliferating and come out from a tissue type. Okay, and starts growing on their own regardless of what other cells are there or contained in that tissue or organ. So these type of cases can also be called uh, metaplasia. So you can see this term metaplasia is a very broad term, and it is used in various type of cell type conversions, various mechanisms of cell type conversion. Whereas the term plasticity, okay, so in general, cell plasticity, it refers to ability of a cell to change its phenotype. Okay, in response to environmental cues. Now note, the cell is not doing it under the pressure of any genetic mutation. So at the gene level, no mutation has happened. Okay. I hope you are able to understand what is genetic mutation. If there is any doubt in understanding this term, please let me know. Okay. So without presence of any genetic mutation, the cell is able to change its phenotype. So cell plasticity is again a very broad term where which people use to again explain change in cell phenotypes and etc. Okay. So here I'm not saying that the cell is changing to another cell type. Here I'm saying the cell is changing their phenotype. So phenotype, you can say that cell initially doesn't have like large ERs, okay, but due to change in the environmental factors, the ER, the endoplasmic reticulum, has become large, okay, in order to support any additional function that is required to sustain in a given environment. So this kind of change is seen. This is plasticity. So these three terms are very different terms, but they can be easily confused with one another. So you have to know what makes these three terms different one another, and then I'm sure you won't be using them loosely ever again. Okay. So if is there any doubt in understanding these three terms, or those who
Ma'am,
have?
yes, ma'am.
Yeah. Yeah.
Ma'am, in trans differentiation, you said right, differentiation of one cell type to another. Does it mean the cell can differentiate to another germ layer cell?
Another.
It's germ layer cell. For an example, if ectoderm cell can be differentiated to a mesoderm cell, is it included like that?
So I can't recall any present example to like give you where this kind of germ layer, like cell of different germ layers have changed, but maybe you can search on the internet. But in theory, it is that is what is transdifferentiation. It can happen in theory, but you might have to look for
Okay,
some
ma'am.
examples, because people are looking into the transdifferentiation properties of these type of cells. It can really help in understanding or in engineering various kind of tissues. So maybe we should look for more examples. One
Ma'am, one
example.
example
Yeah.
I heard it is, for an example, if we get a skin injured,
Mm-hmm.
our skin cells is a ectoderm, right, derivative, right?
Mm-hmm.
So
Okay.
while the dermis develops, already also the blood cells also need to develop, right? So the
Mm-hmm.
blood cells is a mesoderm derivative. So there's a skin cells itself will be changing into blood cells, blood vessels forming cells. Like that I have heard, ma'am. That's what I'm asking, ma'am.
Okay, actually, I cannot confirm this fact. I have to look into it. Okay, because I haven't
Okay, ma'am.
heard
Okay, ma'am.
anything like that. I'll look into it and I'll answer this question in the next class. All right.
Yeah,
Okay.
okay, ma'am. Okay, ma'am.
Are there any more queries related to this topic? Okay, no queries. So we'll move forward. So as we saw, the wound healing process, right? All the four steps. There was first hemostasis, then there will be inflammation, right? And then there will be proliferation of different cell types that will try to close the defect area. And then the remodeling step will happen, which will take months or even years. So that is how the wound healing process happens. So, and in absence of any external factors like scaffolds or any medical agent or anything like that, this is how it normally happens. But we can actually uh, incorporate some external factors to modulate the length of healing, means how long the wound healing process will take place. Okay, so we can do that. So this is called engineering. We are engineering the wound healing process. So in some type of cases, it is like good to delay the wound contracture. So if you recall, in the proliferation phase, the myofibroblasts were there, if you remember, and those were the cells who were responsible for wound contracture, means bringing the ends of the wounds close to one another so that the entire defect area closes, that is wound contracture. And since these cells, the myofibroblast, have the property like partially they are like muscle cells, so they are able to contract. So as I was saying, in some scenarios it is very good to people have observed that if we delay the wound contracture, means if we delay when the wound contracture should happen, then it normally does, the wound healing process will improve. So how people have done it, we will see that in case study 1. So there they have used an analog of ECM, that is extracellular matrix, like a porous scaffold, okay, in order to modulate the wound healing rate of a full thickness skin wound. By full thickness skin wound, I means a wound where both the epidermis and the dermis layer is lost, okay, that is the full thickness skin wound. So they made a scaffold or an artificial extracellular matrix made up of collagen type 1 and chondroitin 6 sulfates. This is the material that they use to make this artificial ECM. And they were fabricated in such a way that they were having a range of pore sizes. Okay, so you can understand what you mean by pores. Pores means the void spaces that are left behind when you know in a mesh kind of a surface, or in a mesh like when the fibers, the polymer fibers, when they are laden one over the other, they leave behind voids, very small voids. These are nothing but the pores, and these are of various sizes. And also they were degraded, they were fabricated in such a way that their degradation rates were different. So what do you mean degradation rate? It means at what rate the entire material of the scaffold will start losing its strength, okay, and will start either breaking down, it will start break down, or dissolute, means you, little by little small fragments of material starts dissolving into the blood or plasma. So in this way, after a few days or few months, the scaffold is no more seen at that wound area. That is the degradation. So most of the tissue engineering application, people want a material, or people want to use a collection of materials which are biodegradable, means which can be degraded in presence of some biological enzymes, okay, that is already present in our body. So they were so this is how they started their study. So these scaffolds were investigated for their ability to delay wound contracture, okay, and promote dermal regeneration in a classic wound healing model. So the classic wound healing model is a square-shaped, full thickness wound where both epidermis and dermis is lost. And these scaffolds that were having a range of pore sizes and different degradation rate, they were studied to see how much they are able to delay the wound contracture, okay, also whether or not they're able to promote complete regeneration of the dermis layer of the skin. So they measured the time required for the wound area to decrease by 50%. So this is the half life, you can say wound half life, uh, t half. So they measured this value. What they found was a scaffold with the pore size of 20 to 120 micron. With the pore size within this range, they were the most optimum pore size, because they were able to delay the wound contracture process by 27 days, okay. So the wound contracture happened after 27 days from the start of the wound, whereas normally uh, it usually happens in 7 days. So here in this graph you can see This is the T half values given in the y-axis, and in the x-axis is the pore size, okay, the pore diameter, average pore diameter. So here it is around 20, and here around 120. You can see the peak, okay, this bell-shaped peak here. This, and in when you see in the y-axis, it is around 27 to 30, like this many days. So uh, this line which I said, that the optimal pore size of 20 to 120 micron, this pore size, this is the pore size range. Scaffolds with this pore size range, they showed a wound half-life of 27 days. Means it took 27 days for the wound area to be partially 50 at least 50% get covered, right? So this is how much the wound contracture has been delayed. Now it is thought that the delay in wound contracture is related to improved healing. Okay, why improved healing? I'll come to that one by in next point actually. So here, uh, it is also correlating to how the fibroblast cells, dermis is made of fibroblast cells. So how the fibroblast cells are migrating through the template or the scaffold. So normally, part of wound contracture happens after the fibroblast cells have covered or spanned the provisional matrix in the wound bed, and then they change to myofibroblast phenotype. Now here, if you recall, in the hemostasis phase, in the first phase, the provisional matrix was formed, if you remember, okay, so where the platelets were trapped and all. So partially the wound contracture actually happens when the fibroblast cells, they have spanned this provisional matrix. Now, if there is a like if we can delay this fibroblast cell migration, then there will be delay in the myofibroblast phenotype change, right? So how this delay is helping us? This delay is helping us first, uh, the inflammation process that is going on, that will get completed. Okay, it will get more time to get completed. All right, the fibroblast cells, normal fibroblast cells, they will be able to cover that area very well, and as well as get more time to synthesize the ECM that is going to form the granulation tissue and all. So before the contracture by myofibroblast will happen. So these processes will help in better regeneration of the dermis layer. Okay, means the dermis structure will the final dermis structure will be more towards the original dermis state that was there before the wound. Otherwise, what usually happens is more and more scar tissue get formed. Means the dermis, in the dermis layer, the cells, the fibroblast cells, they don't get enough time to orient the ECM according to themselves. Okay, and the ECM components, they form mostly scars, which are maybe visible on your skin. So if we can delay this wound contracture, okay, by some engineering method, we can improve the healing process. So as I said, that the optimum pore size of the scaffold that they were using for modulating the wound healing, or for delaying the wound contracture process, was around 20 to 120 micron. So if the pore size was below 20 micron, fibroblast cells, they cannot penetrate. So the average size of a cell is around 10 micron. Okay, say although size of the cell varies from cell to cell type, say it is 15 micron, and your pore size is 20 micron. So obviously the penetration ability of the cell will definitely decrease, right? Whereas if you use very large pore sizes, say 150 or 200 microns, so there what will happen? The cell won't get enough surface area on which it can adhere. Okay, so cell adhesion will decline, And if the cells cannot adhere, it cannot migrate, and neither subsequent cellular fate processes will happen. So we need a optimal pore size in the scaffold in order to delay the wound contracture and thus promote improved wound healing and proper dermal regeneration. Now, they saw how the degradation rate degradation rate of the scaffold is important in this. So they saw that the degradation rate of the scaffold, it should be tuned in such a way that it is approximately equal to the normal time taken for the wound healing to complete. Okay, so it should be approximate equal to that. So the if the scaffold degrades much faster, okay, before the wound healing is complete, then also the contracture in wound contracture won't happen properly. If other happen, like the uh, scaffold is degrading as a very slow rate, okay, then also uh, improved wound healing won't happen. So we need that it is approximately equal. These two time of taken for a scaffold to degrade and the time taken for normal wound healing process, these two times should be approximately equal, or their ratio should be approximately equal to 1. So normally for skin, uh, normal wound healing time, like complete, for all the three processes to complete, it is normally 3 weeks. Okay. So here the size of the wound is not given, means how big size, but I'm assuming that they are telling for the full thickness wounds only. But for bigger wounds, for large wound size, it can vary. 3 weeks can be bit more, whereas for whereas their scaffold that they made with the pore size, optimal pore size, those scaffolds showed a degradation rate of 2 weeks. So 3 weeks, 2 weeks, it is approximately equal to 1. So they saw that when this criteria was fulfilled, they were able to modulate the wound healing rate in such a way both the delay in wound contraction happened as well as the dermal regeneration happened much in a much better way. So are there any doubts in this particular case study? All right, so we'll move on. So in another work, another case study, you can say how they have tried to use the cell based contraction. Okay, means when the cells migrate, they also contract. We will actually see how the cells migrate, means what all steps are involved for the cells to migrate from, say, point A to point B. But in one very important step is contraction. The cells basically contract. So you can imagine how the snails, you must have seen snails, right? So how the snails crawl on a surface or on a ground. So you if any one of you have seen the way the cells crawl, there is one particular point where they contract a little bit. So similar kind of a thing also happens when cells contract on a substrate. So this cell based contraction is used uh, to form tissue like constructs. So cells will contract on a substrate, and this substrate is made by an ECM, or it's a matrix component like collagen, etc. So in this study, so they have incorporated, means embedded, few cells in ECM gel like collagen. And what they saw, so here in this diagram two cases are given, A and B. So in case A, so in case A, what happened is when the cell concentration, when they increase the cell concentration, keeping the ECM compound, that is collagen concentration, constant, when they increase the cell concentration, keeping the collagen concentration constant, the diameter D of the entire scaffold, it started to decrease with time. So you can see the diameter of the scaffold is decreasing with time, because as the cells are growing in number, they are also promoting contraction, means they're bringing the collagen fibers close to one another, thereby decreasing the pore diameters or the pore size of the scaffold. Whereas in another case where they increase the collagen concentration, or they increase the ECM concentration, keeping the cell concentration constant. So here what happened, due to increase in ECM concentration, the diameter, you can say, it increased, the diameter of the scaffold increased, because the cells now are not getting enough, you can say, space for causing contraction. Degree of contraction and the speed of contraction, it has reduced, because of which the diameter of the entire matrix, it has increased. So the same thing can also happen in presence of some compounds or chemical compounds like inhibitors of matrix metalloproteases. So these are proteases or the hydrolytic enzymes released by cells to degrade their surrounding ECM components. The cells usually do that. When a cell wants to migrate through dense ECM, say, matrix, in order to make its own path, they often release these MMP enzymes. So if you incorporate the inhibitors of these MMP enzymes, so these MMP enzymes will not be able to make path for the cells, because of which uh, the contraction motion will not happen. Okay, and because of which the diameter of the scaffold is increasing. So this is how they use the cells, and they grow the cells in the ECM gel, inside the ECM gel, and make tissue-like constructs like dermal constructs, epidermal constructs like this, and then these tissue constructs, they are and they are grafted on some wound. Okay, and then they help in uh, tissue regeneration. So this is another way where they have engineered the wound healing process using the cell, basically cell migration. Okay, cell based contraction is coming from the cellular migration process. So are there any doubts in understanding these two case studies of how they have engineered wound healing process? Okay, so next,
Ma'am, can you
yeah,
summarize it?
okay,
Can you summarize it?
summarize both the case studies together, that is what you are asking.
Yes, ma'am. Yes, ma'am.
Okay, so in case study 1, what they have done, they have checked or investigated how the pore sizes of the scaffold or the artificial ECM is uh, playing a role in delaying the wound contracture process. And wound contracture process is a very small part in the entire scheme of wound healing. As well as they are seeing how the degradation rate of the scaffold, is related to the wound healing process. So they have individually investigated the effect of pore size and the degradation rate of the scaffold with the wound healing process. One is the wound contracture, all right, and also second, also they have seen how the wound contracture and then normal wound healing process is getting changed in the first study. Where in the second study, what they have done, they have used cell-based contraction. The cell-based contraction usually happens when cell migrates. Okay, in order to make tissue-like constructs, the cells are embedded inside ECM gel like collagen. All right, and they saw two cases. In one case, cell concentration was increased, because of which, cell number was increased, because of which the cells contracted, pulled the uh, polymers change, you can say, close to one another, which cause decrease in the overall diameters of the pores. So that caused a change in the porosity of this scaffold due to increase in cell number. Whereas the second case, they have increased the ECM concentration or collagen concentration, and the cell concentration is same. It was given maintained at a constant number. Now here what they saw, cells are not able to contract the ECM that much, because of which you can say that the diameter of the pores remained as it is, uh, remained as the engineered one, or it increased. Okay, or it increased, you can say. You can imagine like that as well if you imagine the reverse case. But if you are engineering a scaffold, you can say that the porosity of the scaffold remained as the engineered one. All right, so here what they are saying in the second study, that how the cellular contraction can change the porosity of the scaffold. All right, and it can actually affect, if you imagine, if you graph this kind of a porous scaffold on a wound area, there the porosity plays a very important role. It actually helps in diffusion of the nutrients and oxygen. All right, so if you are using a scaffold with the high porosity, then the nutrition diffusion, etc., will happen very large. But this but the porosity shouldn't be that much high that it doesn't support cell adhesion. So there should be a balance between or you should have an optimal porosity. Okay, and in second case, you see that the porosity is changed by the cells itself. So the optimal porosity, in the initial case, they were not using any cell. In the case study 1, there was no cells in the scaffold. But in the second case, the cells were embedded in the scaffold. So then you cannot actually, you have to then check what is the optimal porosity, in which after the contraction of the cell, or after the contraction promoted by the cell, the porosity is still optimal for the cell growth. So this is a like they have used this material, like the biomaterial, like collagen, etc., in order to change how the wound healing process is going to get affected. And that too, they're not affecting at a very large scale. Like this process is not affecting every phase of the wound healing. Only small part of the wound healing is going to get affected, very small phase. And that is that they're saying is going to make all the difference. Like they are still trying to understand where we can engineer the wound healing process. And through these two case studies, I tried to explain a few factors through which engineering is possible. All right.
It's got it, ma'am.
Okay. So now let's discuss about fetal wound healing. All right. So fetal wound healing occurs very rapidly, efficiently, as well as perfectly without scar formation, whereas adult wound healing is imperfect and results in fibrosis and scar formation, with poor reconstitution of dermal and epidermal tissues at the healed wound site. So in the case study 1, where they were trying to delay wound contracture, you can say delay in wound contracture can help in proper reconstitution of the epidermis and the dermal area. All right, and also it can result in less fibrosis. Fibrosis is a condition where more and more scar tissue, or only the ECM components like collagen or fibronectin, etc., they are more, and the cell concentration is very less. So this kind of tissue gets formed, and when this happens, that particular area of the tissue is not having, or is not showing the same tissue properties as the surrounding normal one. Okay, so tissue property of that particular area also changes, the strength of that area also gets changed. So we want to, when they are trying to engineer wound healing process, one objective is also to reduce the fibrosis process and minimize the scar formation. Now, even though the processes of reepithelialization, means the formation of the epithelial layer, which we have seen how the keratinocytes they crawl, right? How they crawl over the granulation tissue and try to proliferate, divide and crawl, like divide and crawl until keratinocytes from both the ends meet. So this is epithelialization. So this process of reepithelialization and connective tissue contraction, contraction through the action of myofibroblast. These two processes are common in both embryo as well as adults, but the means by which they occur is fundamentally different. Means the mechanism how these processes occur, they are totally different. In an embryo, how it happens? So in embryo, the gap in the epidermis is closed by contraction of a rapidly assembled actin purse string, while in adult, epithelial cells crawl over the exposed substratum to close the defect area. So this in embryo, what happens? In embryo, what happens? If you have guys have seen a batua, and in batua there is a string that you pull, and because of which your purse gets closed. And everyone is familiar with that kind of a small bag which many ladies still carry, right? So and if you pull the strings open, the bag gets open. So that is the mechanism of how the epidermis, gap in the epidermis gets closed in the embryo. So the actin, you must have heard actin molecules, right? So these are small F-actin molecules that get assembled and forms a string. So when this string gets say contract, when they contract, they it rapidly closes the epidermis gap, and because of which the epithelialization process is complete. The epidermis layer is fully covered now. Whereas in adult, what happens? The epidermal cells, that is the keratinocytes, etc., they crawl, right? They divide and then they crawl, divide and they crawl until the cells meet. The cells meet, right? The contact inhibition process happen. Until that happens, the cells divide and crawl, divide and crawl. So in this way, the gap is closed in adults. So this is the difference in the mechanism of reepithelialization in fetal wound healing or and adult wound healing. And the exposed connective tissue, that is the dermis layer, in the adult, it contains specialized myofibroblasts that contract to bring the two edges of the wound together. Yes, you guys are familiar with that. However, in embryo, the same type of result is achieved by standard embryonic fibroblast. Okay, so here the embryonic fibroblast is acting, and here myofibroblast is acting to do the same thing. Here the same traction forces are applied, means they contract in order to bring the two edges together, but the cell type is changed. In adult, there is an extensive inflammatory response. So neutrophils, macrophages, right? All these, then white blood cells, all these come and they give a very extensive inflammatory response. Whereas in embryo, this such kind of inflammation is practically non-existent. Means it doesn't happen at all. Okay, it is practically non-existent. Now in fetal wounds, here what happened? Uh, Basket weave kind of a structure is there of the collagen matrix. Means if you have seen a bamboo basket, where how the texture is there. So in that kind of a texture, the collagen fibers are aligned in a fetal wound. Whereas in adult wound, the collagens are present as bundle of straws, if you have seen. So in that kind of bundles they are present. So the arrangement of the ECM component like collagen is also different in both fetal wound and adult wound. Then the composition of ECM is completely different in fetus and as well as in adult wound. So In adult wound, you can see that there is fibronectin kind of components that are majorly present, whereas also collagen is also there, whereas in fetal wound majorly collagen type 3 is present. So the composition is also varying, not only the orientation of the ECL, the composition is also varying. So if I were to compare these two type of wound healing processes, adult wound healing and fetal wound healing, so you can like understand through these points. Adult wound healing is relatively slower, whereas fetal wound healing happens very rapidly. It is imperfect, but fetal wound healing is very efficient and causes minimal scarring. All right. Here, the contraction of the wound happens to the action of myofibroblast and epidermal cells. They crawl. This means there is a migration of the epidermal cells. Whereas in fetal wound healing, the wound closes through the action of embryonic fibroblast, and there is a of contraction of the epidermis layer by the actin purse string kind of a mechanism, which I've just explained, like the batua and the string of the batua close used to close the bag. So that kind of action is there that helps in closing the epidermis layer in fetal wound healing. Whereas adult wound healing, there is a high inflammation going on, whereas in fetal wound healing, inflammation is minimal or practically non-existent. Here, epithelial proliferation is very high. Here, epithelial proliferation is very low. Here, collagen is present in bundles, whereas in fetal wound healing, it is present in a basket weave kind of a texture. Okay, and main ECM components like fibronectin, tenacin, these components are there. Whereas in fetal wound healing, there is hyaluronic acid, collagen 3, etc. These are the main components. So, are there any doubts in understanding fetal wound healing and how it is different from the adult wound healing process? So you can now understand, like initially maybe during the wound healing class discussion, I explained that fetal wound healing is a very active area of research. Okay, where they are trying to engineer the wound healing process in such a way that we can achieve few characteristics of the fetal wound healing, like minimal scarring. All right. So less scar formation is kind of uh, cosmetically it is more desirable for people having getting wounds on, like say, face, etc. So this kind of, if people are able to mimic the conditions that promote uh, fetal wound healing kind of features like minimal scarring, then it will be very very impactful for and as well as change the quality of life of various patients. Okay. So another topic that we are going to cover in this class, or also it is also one of the last topic under the section tissue dynamics, is angiogenesis. So if you recall, again in wound healing process, during the proliferation phase, the endothelial cells, they also proliferate, okay, along with the fibroblasts and the granulocytes, etc. So these endothelial cells, they try to proliferate and try to make new blood vessels, okay, and in order to fill up the ruptured blood vessels, all right, and complete a full network of viable full network of blood vessels in the dermis layer. In order to do that, the process of angiogenesis takes place. So what is angiogenesis? Angiogenesis is basically a process of sprouting and configuring new blood vessels from pre-existing blood vessels. Okay, this is angiogenesis, where new blood vessels are formed from pre-existing blood vessels, minded. So there is another process called vasculogenesis, where new blood vessels are formed. It means new blood vessels are formed, and that happens during embryonic development, and also in other adult phase as well. Vasculogenesis has been observed, but the main area where vasculogenesis happens is during embryonic development. Whereas angiogenesis, it happens during wound healing, as I've already said. It also happens during tumor growth, right? So how the tumor is getting nutrients. So they actually try to trigger the process of angiogenesis so that it can get a supply of blood vessels from the surrounding blood vessels that is there in the surrounding healthy tissue. So this when so angiogenesis also happens during tumor growth. So what all steps are there in angiogenesis? So it is a very simple process actually. You will see. First process, this process begins with the release of angiogenic factors. Okay, and these are basically growth factors, and that's why and since these factors initiates the angiogenic process, that's why it is called angiogenic factors. And these growth factors, they are released from the inflammatory cells like macrophages, then platelets, and also ECM matrix. So how it can release from the HCM matrix, you can maybe think over it. Okay, so first they are released, these angiogenic factors are released. These factors are then transported to existing blood vessels in the vicinity. So whatever blood vessels are there in the site which are close to the particular region, so to there the these angiogenic factors, they will get transported through diffusion. Now at the blood vessel, at the nearby blood vessel, these factors bind to the cell membrane receptors of the endothelial cells. So you know, blood vessels, inner lining of the blood vessels is formed by the endothelial cells, and these cells will have some receptors specially for binding to these angiogenic factors. Now, upon binding, what happens? Uh, it initiates degradation of basement membrane. How it will get uh, degraded? So these endothelial cells will start degrading, so start secreting some enzymes, some proteolytic enzymes, which can degrade the protein components of the basement membrane. So after this degradation of basement membrane happens, and endothelial cells also start proliferating. You know, if the new cells need space, right? So degradation of the basal membrane will help, so they will start proliferating and start sprouting. So if we have seen a sprout, how it happens, a small type of a um, like root kind of starts growing, right? Similarly, sprouting, that kind of phenomena happens here as well. And this sprouting, or a very small bulb, very small sprout of blood vessel, is formed by the newly proliferated endothelial cells. Now these newly formed vessels, they will try to extend, they will try to move, okay, penetrate through the tissue. So how they will do that? They will release the matrix metalloproteases, or MMPs. Okay, so they will release these MMPs, which the role of these MMPs is to degrade the surrounding ACM. So when the surrounding ACM is degraded, these sprouted blood vessels, they will try to extend, they will try to uh, penetrate through the tissue. Okay, and while this penetration is happening, new endothelial cells that are getting formed through cell division, they are also they are actually helping this lengthening of this newly formed blood vessels. And finally, these newly formed vessels, the structure of the newly formed vessels, they are stabilized. Okay, they are stabilized by the surrounding mesenchymal cells. So if you uh, go to internet and see the structure of the blood vessels, or what all cells make a blood vessel, you will see that first the endothelial lining is there, then there is a lining of this smooth muscle cell, and also fibroblast, then the pericytes. So these kind of cells, they are recruited in order to stabilize the structure of the newly formed blood vessels. And the entire embryonic process typically happens on the order of weeks. So in this figure, uh, all the 6 points have been covered. Okay, So the first step, as I said, angiogenic factors will get released. Okay, so it can get released by macrophages, etc. Like various sources of are there that can release the angiogenic factors. Then these factors get transported to a nearby blood vessel. So here say this is the blood vessel that is nearby. This is the blood vessel. Now this angiogenic factor has successfully bounded to a receptor, cell membrane receptor, right, of the endothelial cell. This binding initiated what? Degradation of the ECM of the basement membrane. Then it also initiated endothelial cell proliferation. So here the cells are proliferating. You can see this area. The cells are proliferated so much that the mass here, the inner lining, has become bit thick. The cell mass have increased. Then endothelial cell migration is happening or sprouting, means a small uh, area of the blood vessel, it has now started to come out, okay, like a sprout. So this endothelial migration. So here the cells are actually migrating and forming a tube like a structure. Now as this uh, new this particular sprout, or this particular, now this is the new blood vessel that is forming. This has to extend, right? So for the extension, it needs to degrade the surrounding ECM components, and for doing that it releases the enzymes MMPs, or matrix metalloproteases. And as it does it, it grows in length. So it is growing in length, and in order to stabilize the structure of this of this newly formed blood vessel, there are various mesenchymal cells that are surrounding the area in the blood vessel, that is going to get recruited by this newly formed blood vessel. And these mesenchymal cells, they will form a lining, double lining, like it is a multi-lined cells, multi-cellular structure. Blood vessel, as I said, is a multicellular structure. So these mesenchymal cell forms another outer lining to this newly found blood vessel. So after the eighth step, you will form fully mature blood vessels that is able to that can now ready to transport the blood, right, carrying various nutrients and oxygen, etc. So uh, in today's class, I couldn't actually, my health is not very good, so I couldn't actually prepare so much. But I thought we won't be having 2 hours of class, because as I said, my health is not too much. So I had prepared for today's class only this much. If you guys have any queries uh, related to what we have discussed till now, you can ask me. Also, I want to show you guys the portal, because the portal that the ISIS people have made is very good and it's very versatile, and it can actually help me and help you guys to do this course in a much seamless manner. So I wanted to show you the interface a little bit and show you how to log in, etc. So that in future, if any assignment or any quiz, etc. is given, or if you want to quickly access all the course content, you can do it. So if you have any queries, let me know. Then I'll move on and show you guys the portal little bit so that you can access it easily. Are there any queries? No queries? All right. So let me close this screen. I've stopped sharing. So give me a moment. Okay. So you guys can see my screen, right? So if you go to ISIS, this particular link, issonline.in, Okay, so there this kind of a page will open up for you. Are you guys like, have you, if have any of you been able to do it, like open this page in your computer? Can I get a yes or no? Hello.
Yes, ma'am.
Yeah, yes. So I saw that many of you have like don't have the username or don't know the password. So you can actually do it, you know, change your um, username, something like that. So your email id are already registered in the portal, so you can maybe search your username through the email id at least, and you will get an instruction on how to reset your password, etc. So you can do that. Okay, you can reset your password. So I have a password, so through that I will log in. Okay, so this is how the course portal looks like. Now, what they have done now, this ISIS people, they have made two courses by mistake. One is ISIS 060, one is 51. So whatever course material that I'm uploading is 51. So don't get confused. Okay, you have to open the 51 portal. This 61, 60, this ISIS 60, I think you guys are also able to see. But here I'm not uploading any content. Here no content is there, only in 51 the content is uploaded. So let me show you how the portal looks like for you guys. This is the student, this is for you guys. So I've already uploaded week 4 content. Okay, till week 4 I have already uploaded. So you guys can see the announcements here. Okay, what are the announcements are there, and these files are also available to you. So if any of you guys are having difficulty in accessing the files in emails, because often the old emails gets lost, right, in your inbox, because you get lots of emails. So in one go you can download the course content here, from here only. That's a very good thing. So many of you guys had not able to upload the assignment. Okay, so how you guys going to do that? So for that you need to, here when you click the assignment, here click the assignment, here one option was coming, I think, that's add assignment. Okay, so here I'm not sure why it is showing like submitted for grading, but whatever. But I think some add assignment kind of an option was coming, in which you can immediately upload the file and submit. And once you have submitted, this kind of a status will show. Okay, and also you will get an email like your assignment has been uploaded. So here let me show you. I actually tried, I think, to upload one assignment, one file. So this kind of a email also got sent to me directly from the portal that you have submitted assignment 1. So uh, those who have not been able to submit, okay, so you guys maybe can try now. Although I have received it, received your files, uh, I just want you guys to try it. Okay, and if you are facing any particular issue in uh, in not getting a button in your portal, or not understanding how this uh, option works, you guys can ask me. Okay, I'll try my best to explore it myself and let you guys know. Or you guys can also email to the ISIS people. I think they will also reply back. So here, as I'm the teacher, only I can see 3 people have submitted. So this is how it looks for me. Okay, so many of you guys have not submitted, that's totally fine. So here only 3, 1, 2 and 3 people have submitted successfully. I don't know why others could not submit, because it was very easy. And here only I can grade, like I can give you the grades, like the grades will show up immediately here only. I don't have to then immediately individually um, send you the scores, as I usually do, as I've already done in quiz 1 and 2. the your grades will show up in your accounts as well, accounts only. You don't have to or I don't have to do extra work in sending you individual mails. So this kind of flexibility is like offered by the ISSS portal, and I want you guys to explore it bit more. And I've also seen that many of you guys have not even what do you say logged in, okay, not even opened, because as an instructor I get the option to see how many people have opened, how many people are opening the file. If you have even opened the article file, I can know that this many students have opened this particular file. So you should explore the portal a bit more. It will only make things easier. They have made a very beautiful course portal for us. And I hope you guys, after today's a discussion like how I've shown you, you guys will try it, a bit more curious in using it. And if you guys are facing any difficulty, like if you're not able to log in or you have forgot the password, etc., you can reset it easily. You can reset it, and your email ID is your username. Means through email ID everything will happen in your ISIS portal. Okay, so that I'm pretty much sure you don't have to worry about anything else. And yeah, so are there any more queries related to anything that I've discussed? No queries? All right. So let me just quickly take your attendance, and then we'll finish today's class, and then we'll meet as usual on Friday. Okay, Friday and Saturday. In the next class, we will start another new section or new chapter, that is cellular fate processes. What are cellular processes are there? How it happens? Mechanisms involved. Okay, so we will look into that. So one by one, you guys can tell your name so that I can mark it in the attendance sheet.
Ma'am, it's Murli, ma'am.
Murli, another person who just spoke simultaneously, can you repeat?
Shambhavi.
Shambhavi, okay. Next,
Vani.
okay. Next,
Bhupender.
Upendra. Next, next. I have only marked for Murli, Shambhavi, Bhupender, and Vani.
I'm Janvi Sharma.
Janvi Sharma. All right. Next,
Aditya.
Aditya. Okay. So Aditya, you have been missing few classes, no?
Yes, ma'am.
So, all right. Next,
Ma'am Harshita.
Harshita. Okay. Next, anyone else left? Oh, okay. So I have marked for uh, Murli, Shambhavi, Bhupendra, Janvi, Vani, uh, Harshita, and Aditya. Is there anyone else left? I think Vaishnavi is also there, but she is not responding. I saw her name. All right. So, are there any more queries related to anything? Like, have you been enjoying this course, or do you have any complaints, or you want to change something, like a particular thing that I've already shared in the course handout? You don't want things things to happen, or you have any criticism towards my teaching, or anything like if you have any feedback so far, like already 4 weeks have gone, please let me know. So have you guys started interacting with your group mates for the group project?
Not yet, ma'am.
Not yet. I think you should start interacting as soon as possible, because it's going to take time. You know, the task that I have given is going to take time. It's not going to happen in a week or even 2 days. So we do some quality work. That's all. That is why take some time in doing some quality work. Um, but if you are guys like feeling in a difficulty, like people, it sometimes happens that group mates are not very equally contributing, like they avoid something like that. You guys can let me know personally, okay? Or maybe we can do something there. If people are not working equally in the group, maybe we can change the format of the group project a bit, so that it happens much more individual basis. But you guys should let me know. I'm trying to make such environment that we communicate both ways. I'm able to understand your problem and you're able to understand mine in such a way that
Ma'am,
yeah, yes, yeah, yeah.
ma'am, I was unable to attend. Ma'am, I was present.
Yes,
Vaishnavi Mishra.
yes, I have marked your attendance, Vaishnavi. Janvi, you were saying something. You raised your hand.
Ma'am, the day you made the group, the after the next day, we make the group on WhatsApp and a little bit interaction like introduction.
Okay. So have
But
yeah.
not able to work because my exams are like in the sequence manner or like alternative days.
Okay.
Like
Okay.
1, 3, 5, 7, 9, 11 continues my exam, so I can't able to focus on multiple things, but I can manage.
Okay. Okay. It's fine. It's fine. Maybe other group members are also there. They will start working. You can contribute a little bit late. That is why groups are very much helpful. So that's all. If you guys have anything to share, okay, regarding anything, you're not able to understand in particular topic, or you want something more to be added in this course, like you want to learn a bit more about particular topic only, please let me know. Okay, and then let me then wrap up. I'll see you guys on Friday this week. Okay, you guys know can leave the meeting. Thank you for joining.
Okay, ma'am. Okay, ma'am.
Yeah.